aimed to test a potential inhibitor to see whether it can inhibit SHANK3 interaction with RAS. SHANK3 is one of the main structural proteins in the postsynaptic density that creates a scaffold for symbolic synaptic receptors. Mutation or deletion of SHANK3 is associated with autism and other neurological conditions. We have recently discovered the interaction between RAS and SHANK3 via the SPN domain. To investigate the function of this interaction, an inhibitor is needed to carry out a negatively controlled experiment. A potential inhibitor regosative has been described against a similar interaction. It is reported to bind to the RAS binding domain of BRAF and inhibit the interaction between BRAF and RAS. So it is wondering whether this inhibitor can similarly bind to the SPN domain of SHANK3 and then inhibit its interaction with RAS. Therefore, my project is to test this inhibitor against SHANK3 and compare that with BRAF. Based on the structural modeling and sequence alignment, my hypothesis is that this inhibitor can bind well to the SPN domain of SHANK3 and efficiently inhibit its interaction with RAS. To test this hypothesis, I have done the protein expression, purification, and NMR spectrum measurement. I have started with the expression and purification of RAS, SHANK3, and BRAF. All the steps for these three proteins are similar. Firstly, I have done the cell transformation to get the colonies of the transformed cells that incubated at 37 overnight, and then scrapped a single colony to the starter culture, which was subsequently added to the expression medium to express the protein. During the incubation, I have measured the cell density every hour. When the cell density was above 0.6, this solution was then induced with IPTG and incubated at 18 overnight. Then it was ready to harvest the cells and do the bacteriolysis through the centrifugation and sonication. After that, the protein was purified using 5 ml his trap column, and the result was checked by Bradford assay and SDS page gel. The gels were scanned using microwave and distilled on the rocker. The images of gel were scanned using the special gel scanner. Then the purified proteins needed to be concentrated and buffer exchanged to prepare for the NMR spectral measurement. I have tested the binding of this inhibitor to SHANK3 compared with that to BRAF, and then tested the effect of this inhibitor on the interaction between RAS and SHANK3 as well as BRAF. To sum up, the experiment results till now showed that the binding of this inhibitor to SHANK3 was similar with that to BRAF. But it is strange that both bindings are weak, not as strong as reported and expected. Therefore, many further studies are necessary to be carried out to investigate such results.